done with the whole lecture. We will just be summarizing the whole of the lecture to understand what we did and to just recollect the past that you might have forgotten, and then we move on to the part of the exercises. So we started by understanding that cell was the basic unit of life, and without it, sustaining life would be difficult. Then we learned about the word cell was discovered by Robert Hooke in 1665 while he was observing a thin section of cork under the microscope. And this section of cork under microscope was visible like the honeycomb. And in the honeycomb, we know that there are small compartments. So these small compartments, Robert Hooke named them to be cells. And thus the word cell was coined. Then we learned about the types of microscopes. We learned that there is something known as compound microscope that helps you see the cells. But to see the components and the constituents inside the cell that are known as organelles, we required an electron microscope because they were so tiny and we needed a more magnifying power. And that magnification was given by electron microscopes because the magnification power, that means the ability to see the small and the minute particles at a high power, was around 2 to 3 billion times more than a compound microscope. Then we learned about the plant cells and the animal cells, the structure of these two, and we learned that in the animal cell we had the cell membrane. Now cell membrane was nothing but the thin, the flexible wall that was the outermost covering of the animal cell, and it included all the organelles inside it and also the cytoplasm inside it. Then we learned about the cytoplasm. Cytoplasm was nothing but a semi-fluid that was present in the whole of the cell, and it embedded all the other organelles on it, and except the nucleus did not contain cytoplasm in it. Then we learned that the cytoplasm had certain organic ions and inorganic ions in it and components in it. Then we learned about the vacuoles. We learned that in the animal cell, the vacuoles were small in size, and more in number, and these vacuoles are nothing but the empty sacs where the excretory and the secretory products were stored for the meanwhile, that is for temporarily, and then they were removed and eliminated in the whole of the process. Then we learned about mitochondria. We learned that mitochondria was known as the powerhouse of the cell, and it was responsible for converting ATP, making ATP. Now this ATP was made when the food material that was consumed by the organism was broken down and a chemical substance named as adenosine triphosphate, that is ATP, was produced and utilized for the whole activity, cellular activities. We learned about the mitochondria. In animal cells, mitochondria is the powerhouse of the cells because it utilizes the food that is consumed by the organism and converts it to a chemical substance, a chemical product, that is the ATP. Now ATP was nothing but adenosine triphosphate that is used for your cellular activities. We also learned that mitochondria came from a Greek origin, and the word meant it was a granular structure, a small granular structure, and that well quite defines the word. Then we learned about mitochondria having the outer membrane and the inner membrane, and the inner membrane was folded, and on the folded membrane there were cristae, there were structures protruding out known as cristae. Then we learned about the mitochondria having a major role in providing the whole energy in supplying of the energy to the cell, and it being a powerhouse of the cell. Then we shifted to the nucleus. We learned in animal cell that nucleus was centrally located, and it was a very vital organelle that was responsible for carrying out and controlling all the functions that happen in the cell. Just like the CPU was for the computer, then we'll also learn that the structure of the nucleus was porous, and we learned that it played a vital role in the cell division too, and the inherited characters inherited characteristics like your hair color in human beings, like the hair color, the skin tone, etc., or the behavioral characters are also on the inherited characteristic basis, were transferred from generations to generations by the genes carried on the chromosomes. Now, chromosomes were nothing but when long strands of DNA came together to form the aggregate that was resulting into chromosomes, and this is responsible for deciding the characters. Then we learned about the Golgi bodies. In Golgi bodies, we learned that these were flat shaped bodies that were embedded in the cytoplasm of the animal cell, and they contained the enzymes in them to carry out the cellular functions, because enzymes are helpful in acting as catalysts to increase the pace of the function and to enhance that particular action. Then we learned about the plant cells. We learned in plant cells that the plant cells had an extra covering on them that was the tough, rigid, and it was made up of cellulose, though it allowed the exchange of substances through it. And that is known as a cell wall, that was in the shape of hexagonal plant cell structure that I drew. That was the outer rigid covering, was nothing but a cellulose, made up of cellulose, and that was a cell wall, if you recollect that. Then we learned that the cell wall was responsible for giving the cell strength to the plant cell, and it was responsible for giving the plant its shape and its rigidity. And we learned that since we also understand that plants cannot locomote, so this is used as a mode of defense for them. And then we compared both the structures of plant cells and animal cells, and what we found out was that the plant cells had plastids in them. Now these plastids are responsible for storing pigments like chlorophyll, xanthophyll, and other pigments that help in the process of photosynthesis. While in animal cells, these plastids were absent. So this gives us a gist that the animal cells cannot perform photosynthesis while the plant cells could perform photosynthesis. And therefore, these animal cells were not autotrophs while the plant cells were autotrophs, because they produced food for themselves. Then we also learned about the vacuole size. In the plants, the vacuoles are larger and they were less in number, while in animals the vacuoles were smaller and more in number. We also got through this difference by the structures that I had drawn, that the plant cell did not have a centrally located nucleus, while the animal cell had a centrally located nucleus. From here, we shifted our attention to the microorganisms. We understood that what are microorganisms. Then what we came to know was that microorganisms are the organisms that are invisible to the naked eyes. They are tiny, minute, and therefore needed a microscope or a compound microscope for observing them. Then we learned about the types of bacteria. We learned that bacteria were actually present everywhere in the environment, let it be air, let it be soil, let it be water. We then understood about the relationship that we share with these bacteria and how they are helpful and harmful on us. So now the bacteria that were harmful to us were known as pathogens, and pathogens may be defined as the microorganisms that have the ability to produce or to cause an infection or illness in the host. And host is nothing but the organism that is subjected to the pathogen and its actions. Then we learned about the beneficial microorganisms. We learned that in beneficial microorganisms, these microorganisms helped in the processes and made certain products really nutritious, really tasteful and healthy. Like one example I gave you was a fermentation, while the other example that I gave you was in relation to the body. Now in human body, there are a lot of bacteria in the stomach, And these bacteria are useful for maintaining the whole inner environment and maintaining the healthy state of your intestine and your stomach. So this was the one example that I gave to you in relation to human beings and the microorganisms being beneficial to human beings and other organisms like animals. Or to be precise, the multicellular organisms. Then we learned about the 6 types of microorganisms, and these microorganisms were nothing but the bacteria, the virus, the algae, the fungus, then the yeast cells and the protozoan cells. Now in the bacteria section, we learned that bacteria came from a very earlier origin. Therefore they had adapted themselves to form various shapes. The shapes were like rods, that were bacillus, coccus, that was spherical in shape. Then there was spiralis, that was spiral in shape. Spirochaete were curved in shape. And in this way we also came to know that they could exist in clusters or in chains of bacillus and the coccus. Then we learned about in bacteria that bacteria were small, they were like huger than the virus, but they needed a compound microscope to be observed under. So when we observed under the microscope, we could see their shapes and their structures. Then we learned about the bacteria's morphology. Now in bacteria's morphology, we learned that the bacteria did not have a well-defined nucleus. Rather, it had free chromosomes that were suspended in the cytoplasm, and the organelles that were there were only the cell wall, the cell membrane and the cytoplasm. Then we learned about certain kind of beneficial and certain kind of pathogenic bacteria. In the beneficial bacteria, we learned about the azotobacter, that is a kind of bacteria that is helpful for nourishing the soil and is a nitrogen fixing bacteria. Then we learned about streptococcus. Streptococcus was the bacteria that had the ability to infect the food by the poisonous toxin it produced, that was the enterotoxin. And when this enterotoxin when the food was poisoned by this poisonous substance, that is enterotoxin, and was consumed, this would result in the illnesses and these diseases like diarrhea, vomiting, uneasiness, stomach aches, and if this was not cured on time, it would lead to a very aggravated or very acute condition. So it is always prescribed the best to visit doctors and consume the apt antibiotics. Then we learned about the Clostridium. Clostridium was the microorganism that had same effects just like the Streptococcus, but it was found in canned foods, and we also learned that it was anaerobic in nature, and since it was anaerobic in nature, it did not require oxygen for its growth, and hence it would prove to be lethal if it was consumed. So therefore the measures to be taken for this are the food that you consume should be checked for the expiry date, and if any symptoms are observed, they should be immediately treated by visiting the doctor. The next we learned was about the virus. Now in virus we learned that they are really small and particulate microorganisms, and these microorganisms are cannot be observed under the compound microscopes. So what we needed was the electron microscope. Now after observing and seeing it under the electron microscope, what was discovered was that it did not have any organelles or cytoplasm in it. This microorganism, that is the virus, was made up of proteins, and it had only DNA and RNA as the genetic material. And then I explained to you about the mechanism, about how it infected the cells. Then now the full form of DNA and RNA, I'll tell you. The full form of DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Then what we learned about this was that there are certain viruses that affect both the humans and plants actually.
No more spending your energy and money on coaching classes or missing classes for rain, for rally, and any other nonsense. Subjects covered by multiple teachers with repeat telecasts. Doubts, Concepts, applications, no problem. All explained through great and significant animation. So sit back comfortably in your home and watch Study Spectrum TV channel.
Now, these viruses, they can attack and infect both the plant and the animal. So, in animals, the viruses like HIV, that is the human immunodeficiency virus, was responsible for creating a disease as AIDS, that was acquired immunodeficiency syndrome. In this, the immune system of the host is affected, while in plants, the disease that was caused by viruses was like the crown gall disease, because it was found in the leguminous plants' roots, that the roots were swollen because of this virus being affected over there. Then we learned about the algae. We learned that when there are diatoms of algae together existing, they are nothing but unicellular. And we learned that algae were found in wet and damp lands where they had the chlorophyll in them to produce their own food by the process of photosynthesis, and hence they were known as autotrophs. Then we also learned about the fungi, but we learned the same about fungi, but we learned that these fungi didn't have chloroplast in them, because of which they couldn't produce their own food and they had to be dependent on some other organic substance for their mode of nutrition, and hence they are known as parasites. Now, there are certain fungi that are useful and there are certain fungi that were not helpful. These fungi that were not helpful were the ones that created skin diseases and infected some other surfaces. So usually all these are treated. While I also explained to you that in earlier years when the research was really on, there was a scientist named Alexander Fleming. And Alexander Fleming was responsible for the discovery of the first antibiotic, that is penicillin. Now let me just explain to you what antibiotic again is. Antibiotic is a substance, is a product that is created by a microorganism to kill these certain microorganisms that are bad for the health of the organisms. And thus this antibiotic, that is penicillin, was discovered from the fungal growth, that was the penicillium. Now we also learned about the structure, that this structure was filamentous and had hyphae in it. It propagated with the help of spores. Fungus propagated with the help of spores. Then we went on to understand a little bit more about yeast cells. But before understanding the yeast cells, I also told you that not all microorganisms have the ability to produce antibiotics. There are very few who have this ability to produce antibiotics, and the ones who have, we are using them today for treating our various diseases. And this has set a benchmark in the medical history in the field of science and has proven to be really helpful. Then we learned about the yeast cells. We learned in yeast cells it was very helpful in the process of fermentation. It gave us certain desired and tasty and very nice textured products like the dahi, the bread, the dosa batter that you have, idlis, and there were certain fermentations that were non-desirable. Now these fermentations that were non-desirable would lead to certain unwanted fermentated products that wouldn't be consumed. If consumed, it would cause illness. So this would actually produce a very off smell in it. Then we learned about protozoans. We learned that one example of protozoa was the amoeba, and it had only a cell wall, a cell membrane and a nucleus. It just had a cell membrane and a nucleus in its structure and nothing else. Now, if this amoeba is actually not a very healthy protozoan. Now, once it is consumed, it produces the same stomach upsets and diarrhea, vomiting and dizziness and other factors. In order to avoid that, again cure and treatment is sought and it is needed on time. So now there are certain protozoans that are actually helpful also, while there are also certain algae which are worth being fed on. for certain herbivore animals. But also at the same time, I also told you that if certain things have a good side, they also have a negative side to that. So when this algae is grown in tanks that are stored above your building, so what happens is it would produce a very off smell and very unwanted smell from it. And this is not desired, so hence precautions should be taken and cleanliness should be kept every while. So now we are done with explaining. I'm done with explaining you about the summarizing whole of the lecture. Let's just go to the exercise. The first exercise that we have is a very interesting one. It's the multiple choice questions. Now in multiple choice questions, the first one that we have, I'll be giving you the answers. You have to figure the answers out onto your own selves also. And you have to try to relate every single option that is given. So the first one is the dash is the organelle found only in plants. Is ribosome the organelle or is mitochondria the organelle? No, mitochondria is not the organelle because we learned that mitochondria is found both in animals and in plants. So it can't be an exception. Is plastids? Yes, the plastids is the answer because plastids have chlorophyll in it, and this chlorophyll helps plant to produce their own food, while animal cells, as I told you in the summary, also have no chlorophyll in it, therefore no food can be produced by animal cells. So the answer for this is plastids. And the last option is the nucleus, but the nucleus is present actually in both animal and plant cells. So let's move on with the first exercise that we'll be doing, that's the multiple choice questions. So what we have here is the first fill in the blank. Now I will be dictating you the answers, but you also have to try to reason out the reasoning of the choice that you make. So the first one is the dash is the organelle found in the plant. Now is ribosomes the one, mitochondria, plastids, or the last option, that's nucleus? Well, mitochondria and nucleus, we know both are found in the animal cells too. So it cannot obviously be the organelle that's only found in plant, while ribosome is not an organelle at all. So plastids, yes, of course plastids are the answer because plastids contain pigments in them like chlorophyll and xanthophyll that are responsible for giving the color to the plant and are also responsible for helping the plant in the process of photosynthesis, in which it is able to produce the food for itself and meet its nutrition. So the answer is C, plastid. That's the answer. So I'll just write that down there. Let's move to the next. That is, chromosomes are found in the organelle called dash. Called what? So the options now we have are plastids, A plastids, B mitochondria, C nucleus, or D Golgi bodies. Now let's try to read the question once again and reason out why. Chromosomes are found in the organelles called dash. What did I explain to you? With the reference of the plant cell and the animal cell. Now, do you think plastids are the one that are responsible for carrying chromosomes? Well, that's not, because it just contains the pigment that gives colors to the plant. Mitochondria, no, it's the powerhouse of the cell. Nucleus, yes, because nucleus is responsible for having chromosomes and helping the cell-in-cell division. So the answer is the chromosomes are found in the organelle called the nucleus. The option is C, nucleus. The next one that we have is the dash cell carries out photosynthesis. The options we have A, plant cell, B, yeast cell, C, muscle cell, or D, nerve cell. Now the answer is quite obvious. It is obviously plant cells because the yeast cell also does not have any chlorophyll in it. So the answer is A, plant cell is responsible for carrying photosynthesis in it. Now I'll just quickly repeat it all to you. The first answer that we have is the plastid is the organelle found only in plants. Of course, because the plastid has chlorophyll in it, and chlorophyll is only found in plants. The next one is chromosomes are found in the organelle called the nucleus. Chromosomes are the genetic material that are responsible for giving the character to every individual living being on this planet. And therefore, chromosomes, we also understood that chromosomes were found in nucleus. Why? Because nucleus plays a vital role in cell division and passing on the characters that are passed to the next generation. So the answer is chromosomes are found in the organelle called the nucleus is the answer. The dash, that is the plant cells, carries out photosynthesis. Now this is a very obvious answer, and I've been explaining this to you throughout the chapter, that the plant cells are responsible for carrying the process of photosynthesis, and hence we are also dependent upon plants because we cannot produce food for ourselves, and we depend on other modes and other sources for the nutrition that we require for our daily activities and daily purposes to sustain life. So let's move on to the next exercise. That would be just fill in the columns and fill in the columns will have no MCQs, so we will move on with the next section. The next one is the name cell was first coined by the scientist. Now I remember telling you in the introductory part that the name of the scientist was Robert Hooke, and he was an English scientist. So the name of the scientist is Robert Hooke. Let's proceed to the next one. That is the cell wall of the plant cells consists of the substance dash. Now I explained to you that the cell wall was a hard, rigid, and the structure that gave strength to the cell, and I mentioned a substance that it consisted in it. Do you remember that substance? That substance is actually cellulose. So because of cellulose, it gets its cell strength and the perfect shape that it has now. So we'll write that down too. Cellulose is the answer. The next one is the cell wall is found only in the dash. Now it's obvious that I explained to you about cell wall. We're having cellulose as the material in it, and it's being seen only in plants. So the answer over here 2 is plants. Also, when I explained to you in the difference, I told you that there is a cell wall that is an extra covering in the plant cell, and the animal cell has only the cell membrane and nothing else. So the answer is the cell wall is found only in the plant cell over here. The next one is this dash is the powerhouse of the cell. So just try figuring it out. What is the powerhouse of the cell? How many organelles did I teach you? I taught you about the Golgi bodies. Do you think that's the answer? No, that's not, because it has enzymes in it. The next is vacuoles. They are just the empty spaces. So that's not the answer too. The third one was about nucleus, but nucleus cannot be the answer. The fourth one is the plastids. No, that can't be the answer too, because that's found in the plant cells. Then what's the answer to this? The answer to this is the mitochondria. So mitochondria is the powerhouse of the cell. So we'll write it down that mitochondria. You spell it to be M-I-T-C-H-O-N-D-R-I-A, mitochondria. The last one is the cell membrane is dash. Now there can be 2 to 3 answers for that actually. The cell membrane is flexible, the cell membrane is thin layered, and the cell membrane actually gives shape to the cell. So we'll go with the first 2 options, that is the cell membrane is flexible and thin. So I'll write it down, flexible and thin. So now we're done with the fill in the blanks. We'll again repeat it for you to be thorough with it. You need to repeat the answers with me. So the name cell was first coined by the scientist Robert Hooke. Robert Hooke was the first scientist who coined this name cell. I told you how he discovered it. He discovered it by observing the section of cork under microscope. He found small compartments in it, and these compartments were named as cells. So the next one is the cell wall of the plant cells consists of the substance cellulose, because cellulose gives it its strength and its hard shape and structure. The next one is the cell wall is found only in the plant cells over here. That's very obvious answer because we just solved one question about it that had the same answer actually. Dash, that is the mitochondria, is the powerhouse of the cell. I explained to you why, because it takes the food that is consumed, breaks it down and converts it into a chemical energy known as the ATP, that's adenosine triphosphate. That's A, that's the T, and this is the P. So that's ATP. Then the last one is the cell membrane is flexible and thin. We studied about this. Now let's move on to the next exercise. So the first question and answer we'll be discussing about is list the characteristics of the cell membrane. Now I think I have made you a little thorough with this because I've explained and I've repeated this same set of answer 2-3 times for you, but still we'll repeat the points again. Now the characteristics of the cell membrane are that it is the outer covering of the cell. As I told you, it's the outer covering of the animal cell and the inner covering of the plant cell. So this cell membrane is the outer covering of the cell. It is a very thin, flexible layer. It is a very thin and a flexible layer, and the cell membrane protects the inner part of the cell and regulates the flow of substances that enters into and pass out of the cell. So we'll repeat this answer again. The characteristics of the cell membrane are that cell membrane is the outer covering of the cell. It is a very thin and a flexible layer. Then the cell membrane protects the inner parts of the cell and regulates the flow of substances in and out of the cell that enter the cell and pass out of the cell. The next one is that the cell membrane maintains the shape of the cell. As I told you earlier and we discussed earlier too, it maintains the shape of the cell. So let's move to the next question, that is explain the functions of the nucleus. The functions of the nucleus can be explained by the 2 points, and those 2 points are the main functions of it. The first one is the nucleus controls all the functions of the cell and takes part in cell division. Now I remember telling you that cell is continuously multiplying and replicating. This is by the process of cell division. The layer of skin has millions of cells on it, so imagine this is a very rapid process and it keeps on happening to form these long layers of skin on you. So the nucleus controls all the functions of the cell and it takes part in the cell division. The next one would be the inherited characteristics are transmitted to next generations according to the genes on chromosomes in the nucleus. So we'll repeat this answer once again. The nucleus controls all the functions of the cell and takes part actively in cell division. The next one is that the inherited characteristics are transmitted to next generations according to the genes on the chromosomes, and these chromosomes are responsible for giving us characters, that is the inherent characteristics. That means the qualities that you have in you, like your color of skin, your hair color. I've explained this to you all earlier. Let's move to the next question. So, what functions do vacuoles perform? We discussed in the section of vacuoles while we were discussing about organelles that vacuoles store the products of excretion and secretion temporarily. So, the only function that vacuoles perform in plants as well as animal cells are that vacuoles store products of excretion and secretion temporarily. Let's go on to the last and the final question of this lecture, that is, elaborate on harmful bacteria and microbes. So, I explained to you about what harmful bacteria were. Harmful bacteria were that had the ability to create illness or disease or to deteriorate the substance that it infected on. So the main example of the harmful bacteria, we studied about two. That was the Staphylococcus bacteria and the Clostridium bacteria. So in Staphylococcus bacteria, we learned that it produced a poisonous substance called as enterotoxin. And when this enterotoxin entered the food, it made it poisonous too, and by chance if this food was consumed, it would finally result in giving infection or disease to that person who consumed it, or an organism or an animal who consumed it. So in this way, Staphylococcus bacteria proves to be harmful. And the other one is the Clostridium. It over here. Now Clostridium produces a poisonous substance too, that is seen in the canned foods, and I have repeated a couple of times for you that Clostridium bacteria has the ability to grow in an anaerobic condition, and because of which it becomes a very lethal microorganism. Now, let's not talk about only bacteria and their effects on the health. Let's also talk about protozoas and yeast cells for that matter. Now, just not bacteria are harmful, whereas just the protozoas and the yeast cells too are harmful. So the protozoas like amoeba are harmful, and they cause various illnesses once they enter the body through the food they have infected or through any other medium that they have infected, and they enter the body to cause vomiting and diarrhea. So for all these symptoms, it is really important, let it be the Staphylococcus bacteria's symptoms or the Clostridium's infections or anything that is infecting our body, it is very important to go and visit a doctor as soon as possible and get right medications and antibiotics suggested for that, because if a little bit more delay is seen in this, that would lead to nothing but worsening the case and making it more severe. The next one is, though it's not really harmful, but it's not acceptable either, that's the yeast cells. Sometimes it produces a very unwanted fermented products by the process of fermentation. And these are most times they result in the materials that are not palatable at all to be consumed, and hence needs to be discarded and not consumed for that matter, because nothing that is unpalatable for us will always result in proving harmful for the body. So taking care of health, being prescribed to proper antibiotics on time is a very important part, and having your regular visits to doctor is really important. So I'll just repeat these two questions and then we'll be done with the session. So the third question is, what functions do vacuoles perform? Now, vacuoles store the products of excretion and secretion temporarily. This is the only main function that it performs. The next is, we had to write a note on harmful bacteria, but I also mentioned certain other microorganisms that were harmful. So we understood that harmful bacteria was nothing but the bacteria that had the ability to cause disease and to infect the food or any other organism that it had entered in. So the two examples that we considered were Staphylococcus bacteria and Clostridium. Now Staphylococcus, the name here, coccus itself mentions to you that it is spherical in shape. The bacteria shape is spherical. And Staphylococcus bacteria does nothing but it produces a poisonous substance, that is the enterotoxin. And when this enterotoxin is introduced into food and that food is consumed by any other organism, other cellular organism, it would lead to giving them an infection of or disease like diarrhea or the vomiting or stomach aches. And in order to reduce these or to cure them, it is very important to visit doctors at the right time and have right prescriptions. The next example was Clostridium, that was produced in canned foods, and it was, since the canned foods are very compressed and have no oxygen in it just to keep them stored for a very longer time, so there are chances that Clostridium bacteria might occur as the date of expiry is passed on. So it is very important to check the date of expiry and then consume foods. Then the next microorganism was the protozoa, in which amoeba is a harmful microorganism that has the ability to infect the human beings or any other organism and create diseases like vomiting and diarrhea. While there are certain kind of yeast also that produce a very unwanted type of fermentation, and most times this results in nothing but very unpalatable consuming materials that cannot be actually consumed. So that was it for today. I hope most of the doubts on the structure of cell, the components of cell, and about microorganisms was cleared. And please stay tuned to Study Spectrum TV channel, because there's a lot more coming up for you to gain more knowledge about and to be interested in about. Thank you.